Okay, sorry. Okay, good afternoon, everyone. Also, good morning and good evening, depending on the time zone from which you are joining. Hope everything is in place with respect to the systems. A warm welcome to everyone. I am Sujit Nair, the co-founder and director of Spectratix Pharma Private Limited, and I have the privilege of serving as the host and moderator for this session. Before we begin, I have to briefly introduce Spectratix Pharma. Spectratix Pharma is headquartered in Pune, Maharashtra, and we was established with the vision of delivering a high quality services in the pharmaceutical and biotechnology sector. Our organization is committed to supporting the development of quality, safe, and effective medicines that ultimately will improve the patient care. Through our academic and training division, which is known as Pharmacan Academy, we strive to bridge the gap that exists between academia and industry by offering different industry-oriented training programs for researchers, students, and also industry professionals. Our activities include online, offline certificate courses, hands-on internships, industrial training programs, and also research support and academic project guidance. We are also very proud to support the research work of nearly around 10 PhD scholars who are currently conducting their research activities through our laboratories and also the associated facilities. As an extension of this commitment to knowledge sharing and professional development, we launched this Spectratix Knowledge Series. Through this initiative, we invite distinguished experts from academia and industry to share their expertise in, or rather, on emerging scientific developments and contemporary topics that will ultimately contribute to the academic growth and also the professional advancement of all our participants. I am very happy and delighted to mention that today marks the 22nd session of this series. And it is also a great pleasure to introduce our esteemed speaker for today's webinar. We are really honored to have with us Dr. Uma Chandran, who serves as the CEO and founder of Scinome Consulting and Training Services. She is an accomplished omics scientist, a bioinformatician, and an entrepreneur, and she is dedicated to advancing the personalized healthcare and wellness through this omics driven research and innovation. Dr. Uma Chandran completed her PhD in biotechnology molecular reproduction from Rajiv Gandhi Center for Biotechnology, Kerala, and subsequently pursued her postdoctoral research as a DS Kotari Fellow in network pharmacology at Savitribai Phule, Pune University. Through Scinome Consulting and Training Services, she leads the initiatives that transform complex omics datasets into meaningful biological insights, supporting cutting edge research, product development, and translational science. Her work reflects on strong commitment to bridging biology and technology while promoting precision health through evidence-based scientific approach. Her major areas of interest include biomedical research using omics data, personalized wellness through omics or genomics rather, drug discovery inspired by Ayurveda and traditional medicine, genetic and genetics counseling. Through her remarkable academic journey, scientific expertise, and entrepreneurial vision makes her an ideal speaker for today's session. And we are really privileged to have her with us. Thank you, ma'am, for joining us. On behalf of the entire team at Spectratix Pharma Private Limited, I would like to extend a heartfelt welcome to you, Dr. Uma Chandran, ma'am. Thank you for accepting our invitation and also for taking the time to share your knowledge and expertise with all of us today. Ladies and gentlemen, please join us in warmly welcoming Dr. Uma Chandran. Ma'am, the platform is now yours. We look forward to an insightful session. Thank you so much.
Thank you, Dr. Sujit, for this nice introduction and for inviting me to deliver a webinar on AI meets network pharmacology. So, and good afternoon to all. So I think let us begin the session. Let me share the screen. Is my screen visible to everyone?
Yes, ma'am. Clear.
Okay. So, as Dr. Sujit mentioned, I represent Scinome Consulting and Training Services. So, we provide omics data consultancy services and bio IT training services. So today, when he asked me, so if he can deliver a webinar, so I thought, okay, so network pharmacology, so that is my major domain. So I thought I will do a session on AI meets network pharmacology, that is reimagining drug discovery from phytochemicals. I think the slide is not moving. Can you see my slide?
It's on the first slide, ma'am.
Okay, so I'm not able to go back as well. Let me check. So is
Yes,
it
it's
moving?
moving.
Yeah. Okay, so this is the webinar flow. So we will be starting with the introduction. So we can have a quick look at the drug discovery crisis. Then we can look at the fundamentals of network pharmacology, followed by a network pharmacology workflow, and how we can see how we can integrate this artificial intelligence in network pharmacology. Then if we can integrate this AI, then what would be the AI network pharmacology architecture, and we can also just see what are the future directions. So let us start now. So we all know that in pharma, so the drug discovery crisis, the scientists they face. So we know that the drug discovery, so that is a long time process. That is a very time consuming process, and there is a lot of fund going in that one. But even after that much of this long term effort and this much of this this large investment, there are a lot of this failure rate. So if we look at the statistics, it says that 90% failure rate. So we are witnessing in the conventional or classical drug discovery. So that is where we say that so there is a big crisis in the drug discovery. That is the classical drug discovery. So that does not mean that we are this not efficient at discovering molecules. We are very much efficient at discovering molecules. Then where are we failing? So otherwise, where did we fail? So we failed at discovering medicines. And so why? So what was the reason behind that failure? So why those drugs fail? When they investigated, so when they retrospectively checked, so they found that actually, so the problem was there in the approach. Actually, so for that developing the process, so every that process, for example, we know that actually in industry, so that is very much process oriented. So in order to develop a process, even though the complex biological system is very much complex, so to develop a process, we look at only one angle. So where did we look at? So we looked at the single target approach. That is, if there is a condition, so then the thinking was that actually one target would be affected, and if we can correct that target, then we can achieve a solution for that. So that was the very linear thinking, and actually we ignored the complexity of our biological system or the complexity of our this disease biology. So, and there we can say that, or I realized that the failure is not in the drug, but it was in the model they used to design it. So that is when they thought of this thinking the shift in that drug discovery. So what can be done to this address that kind of an approach, or how can we develop that kind of a refined approach to address these issues? So that means actually we were following a reductionist approach. So then that reductionist to systems biology, a systems biology approach is required. So that there we realize that that systems biology approach is required. So what is a systems biology approach? That means that actually in reductionist biology, we try to understand the part. So, and we know that understanding the part is not enough. So we need to understand, or we must understand the interactions. So we know that the systems, the complex system, our biological system, that is a very complex system. And so that is when the emergence of network pharmacology happened. So this happened some 15-20 years ago. So this, what is this network pharmacology? So this network pharmacology, that is an approach so that integrate multiple disciplines. So rather than looking at one aspect or this one area, this network pharmacology is integrating multiple disciplines to understand how drugs interact with biological systems. So this integrates mainly three systems, that is systems biology, pharmacology, and bioinformatics. So this network pharmacology explores how drugs influence the entire biological networks. And actually in between this just want to say, so if there are any questions to be discussed, we can discuss in between, or Dr. Sujis, what is the pattern? So do we need to discuss the questions afterwards or in between?
They can ask in the chat box also, so that the flow is not lost. So we'll take up the questions towards the end.
Okay, yeah, that's perfect. Okay. So then, why this network pharmacology? So I told you that there was an emergence of this network pharmacology. So then, why they prefer or they preferred that network pharmacology? Because this network pharmacology that look at the diseases as the network perturbations. So we looked at that actually this biological system is a very complex system. So that is acting as kind of a network, and if there is a perturbation that can be due to this multiple targets or might be due to a key target. So when that target is perturbed, so that is sending that perturbation signals to the downstream targets. So the whole network is getting disturbed. So the diseases, those diseases are mostly due to the perturbation of such network. And network pharmacology looks the drugs as the network modulators. So in network pharmacology, the goal is to restore the system balance. So that is how these perturbed targets are to be this modulated or balanced to attain the this proper balance that was there earlier in the normal condition. So that is the main goal of this NB, network pharmacology, that is to restore the system balance. And why we do is that actually these complex diseases that require the network level solutions, not the single target fixes. And now we can look at the core principles of this network pharmacology. So there are three core principles for network pharmacology. The first principle, that is the polypharmacology. So this polypharmacology, you might be knowing that one, that is, so one drug that is, or one compound, so that is not may not be acting on one target. That would be modulating multiple targets. So that is the principle of polypharmacology. So by modulating multiple targets, they can simultaneously modulate pathways. And this pathway, simultaneous pathway modulation, that reduces resistance and that improves the efficacy. So these drugs also, they do not act in isolation. So they do not go and bind you only on one target. Rather, they modulate multiple targets and they modulate that network in such a way that actually when we have to design a drug based on this polypharmacology approach, we will be designing a drug in such a way that so that is modulating multiple targets and bringing back that balance of the network. Then the second core principle of network pharmacology, that is the network biology. So in network biology, so we know that these targets, they exist in systems. So that is, if you look at the protein, so these proteins, they are not acting in an isolated way. So if we look at the receptor, so if there is a this cell membrane receptor, so we know that actually when a ligand is binding to that cell membrane receptor, So that is passing signals to the cell, in the interior of the cell, and that is initiating the cascade of a reaction. So that means that there are a lot of proteins involved in that this cascade of reactions. So this network biology, so that is the principle of that network biology is that these targets, they exist in systems, and this, for example, we know that this protein interaction networks. So these proteins are this coming in that network, and this one protein is sending signals to the another one. So that in turn sending signals to the downstream targets. That is how that acts. So and in that cascade of reaction as well. So one target in between, if that is affected or that is disturbed, so that entire cascade of reactions will get affected. And there are in our biological system, there are proper this feedback loops also to maintain the balance of that system. And the third core principle of network pharmacology, that is the emergent effect. So the emergent effect means the system response. So here in this picture, you can see that actually this is kind of a network representation. So in that network representation, so if you look at this each nodes, so that would be acting each other. So when they act each other, so there will be some micro level reactions. But when it comes to the macro level, the cumulative effect or the emergent effect we get, that would not be similar to the that interaction between those two nodes. So what we get as an outcome, that would be kind of an emergent effect. So the emergent properties, so that is the this key aspect of this emergent effect. So that include the whole that is not equal to the sum of the parts, and so there will be a system level behavior, and this causes the this non-linear responses. So that drug effect, that is a network response, that is not a this target one, that is not a this linear this response. So that is the third core principle, says that is the emergent effect. Now let us look at some network terminology. So you may be familiar with this network terminology, but if anyone is there who do not know about this network terminology, so this is kind of a this refresh for others, and those who do not know, so they can also this know these terms. So knowing this network terminology, that is also very much important. So in network, so the node, so that represents the entities, each of the entities. Say here you can say a lot of this round bodies, those are known as the entities. So that can be gene, protein, pathway, compound, or plant, anything. So anything we represent, so any kind of entity we represent in the network, that we call it as the node. And edges means so the connections between those two nodes. So this, and actually there are meaning for these connections. We cannot simply draw this connection. So that connections, that also has some sign based on some scientific evidence or some this biological connections. We make these connections, and those connections are called as the edges. So that denotes the interaction between the nodes. And the degree, so degree means the number of connections. So for example, this one node, that is connected to how many nodes, so that represent the degree. So this, there are a lot of this topological terms. This degree is one such topological term. And next one is the hub. So hub means the highly connected node. So here you can see this is represented as the hub gene. So that means that actually, so even though this nodes, all the nodes are important in a network, but some nodes are very much important compared to other nodes. So those nodes are known as the hub nodes, or this, if that is genes, we call that as the hub gene. So and more, these hub genes or hub targets are very much important in terms of drug discovery. And modules are the functional clusters. For example, so when we have a network, so we can also see whether there are some cluster formation based on some functional aspects. So we call such types of clusters as the modules. So these are the key terminologies one should understand, or one should know, or this when they deal with the network pharmacology. Now, we this started from the classical pharmacology, classical drug discovery, and how did we reach to network pharmacology. And so that means that network pharmacology, so that there was that target centric approach, and then we reached to the network thinking approach. So in that target centric approach, the question asked was that how does this drug do to one protein? Then we were focusing on single target interaction. So that was giving us a linear cause effect assumption. But in network pharmacology perspective, so we are asking these questions. That is, how does this drug affect multiple targets, and how are these targets interconnected? What is the system level response? And here in network pharmacology, so what we actually study is that we study the changes in biological networks, and we study the modulation of pathways and signaling cascades. So that is how we study the overall disease network rebalancing. So that is what we actually study in network pharmacology. So that is in network pharmacology. So we know that drug action is not an isolated event, rather it is a network-wide transformation. Now, let us look at the phytochemicals. We know that this phytochemicals, these are very key. This compounds, this kind of this compound libraries, or this phytochemicals or natural compounds have contributed a lot to the drug discovery. So why these natural compounds are very much important? Because these natural compounds are the evolution optimized molecules. So these natural compounds are designed over millions of years of biological interactions. We know this plants, they mainly produce this phytochemical. Phytocompounds are produced by these plants. So these plants, they cannot move from one location to another. So in order to have their survival, so they have to produce some compounds so that protect them from their surroundings and their this interactions with other bugs or insects and everything. So so that is why we call that as those are the evolution optimized molecules. And as these are evolution optimized molecules, so they show high chemical diversity, and those are the rich source of bioactive compounds, and they inherently have this kind of a multi-target activity. And if you look at, so we have been studying these phytocompounds, right? So if we look at those phytocompounds, we know that actually they are not designed to act upon a single target, but they can modulate multiple targets. And so because of that, they are better suited for complex diseases. These complex diseases, or they are caused by perturbations of multiple these targets. So to address that complex diseases, these phytocompounds or phytochemicals are better suitable ones. So this otherwise we can say that as phytochemicals are the network modulators. So suppose if there is a disease network, so that is showing perturbation due to some key targets, and if we can test some phytochemicals, if that is having the capability of modulating these targets, then so we can say that okay, so this phytochemical can act on this multiple targets and balance or bring back the balance of the network. So that is due to their multi-target activity. So not it is each and every phytochemical, they may not be acting on multiple targets, but mostly the phytochemicals are having that kind of a capability. So when we do the in silico experiments also, we know that this when we do the molecular docking experiments also, we know that actually these phytochemicals, they can have the capability of this binding to pockets of this multiple targets. And these phytochemicals, so they are acting by fine-tuning the gene and protein interactions. So once they bind at that this particular target or multiple targets, so they this modulate the pathways associated with the targets, and they this regulate the this inflammatory or metabolic modules. So in that way, they this act at the network level, and through that action, they shift the system from the disease state to the balanced state. So that is about, I think, so we have set the background for this network pharmacology, and also this importance of phytocompounds, exploration of the phytocompounds for this maintaining the balance of the perturbed network. Now let us look at the network pharmacology workflow. So now as the AI emerged, so we can call this as the conventional network pharmacology workflow. So this is the overview of this network pharmacology, this workflow. So when we deal with network pharmacology, we start with the chemistry, and this we end up with the this mechanism for that this phenotype modulation. So the compounds, we start with the this phytocompounds. If we are dealing with the this Ayurvedic formulations or this botanicals, we start with the compounds present in the botanicals or the this plants contributing to the formulation of that this Ayurveda formulation, which we are taking for the study. So we start with that compounds, then we find the targets of those compounds, then we find the disease gene. Suppose if we are studying a particular disease, we collect the target information for that this disease genes. Then we do the overlap. That is the this first layer, that is the compound target, and the second layer, that is the disease target. And we do the overlap analysis, and that overlap, that is the targets that is coming at this region, at the junction, we call that as the targets associated with their disease, so that are potential to be modulated by the compounds that we started with the study. So then we do develop the network. So then we identify the hubs, that is we do the network topological analysis, and we do the hub identification, then pathway or functional enrichment analysis, and the validation. So this is the overall workflow of the network pharmacology. Now let us also visit some of the databases which are very essential for getting this information. So that is in conventional network pharmacology, these databases are very much important. So we are getting that information, that the basic information to develop a network from this biological databases. So for each type of this, each layer of that knowledge, so we have to depend on various databases. So let us explore the for compound identification. What are the databases being used? So IMPACT, that is an Indian derived database that gives information on Indian medicinal plants, their phytochemistry and therapeutic information. And PubChem database, and there is Dr. Duke's phytochemical and ethnobotanical database. And this PubChem and Chembel we can then utilize for the profiling of the whatever compounds we have identified through this even traditional Chinese medicine databases are also available. So from whatever databases we have collected the compound information, to have a profiling of those compounds, we can utilize the databases such as PubChem and Chembel. Then, so once we get that compound information, we can do the ADME screening. So this ADME screening evaluate the bioavailability, drug likeness, lipophilicity, molecular weight, all those characteristics of the compound, and also see this whether that is a druggable compound. So and we check the Lipinski's rule of 5 also. So for that, this online this database or resource that is known as the Swiss ADME, so that can be used. So throughout this session, I'll be discussing the open source databases or the sources that is available for performing the network pharmacology task. And for target identification, so there are multiple ways to do a target identification. So one is directly extracting from the databases. So that is, so there are databases that give connections between the compounds and targets, and we can extract the information of the targets from those databases. Otherwise, we can do the predictions, target predictions using tools like Swiss target prediction. So where they do that is, if we submit a compound, that is the 3D structure or 2D structure or SMILES information of a compound, then that matches similar compounds, and if that compounds are having some target information, so that gives the predicted targets as the compound targets. So that is how such databases or such resources work. So there are multiple ways also to do that one, as I told you. So we can utilize such databases. Then for getting the disease gene collection, so that is, as I mentioned, suppose if we are studying a disease, so we need to gather information regarding the disease genes or targets associated with that disease. So for that, there are many databases like gene cards, open targets, disgenet. So we can source that information from such databases. And then the next step in that process, that workflow, is the overlap analysis. So for that overlap analysis, so we can use some sources like this Venni 2.0, that is that online tool available. Otherwise, we can use this Google Sheet or Excel sheet, VLOOKUP formula or any other formula. Even some Python scripts are also being used to find the this common targets between the drug targets and the disease genes. Then once we get that information, that is the compound target information and the disease target information and the this common target information, then we will have to find make the final compound disease associated target information. So using that information, we can develop the network. So for developing that network, so this software called Cytoscape can be used. So this is a representative this network, that this is the botanical bioactive target network of Triphala. So we had published this in one of our previous articles. So after developing that network, actually here you can see, so this network, when we develop this network, so that is not only for visualization, so that is for analysis as well. So we can do the analysis based on topological parameters of a network. So such topological parameters are the degree centrality, between the centrality, or this neighborhood, this actually neighbor, how many neighbors are there, or some this there are multiple algorithms for there to identify the hub targets. So there are multiple ways for that one. Eigenvector centrality. So there are many topological parameters we can analyze. So the whole objective of doing this topological parameter evaluation, that is to identify which are the key genes in the network. So because actually, so that key gene identification or hub gene identification is very much important, because so that would be the highly connected gene or node in that network. So if there is any perturbation on that node, the entire network will be perturbed. But if that is not an important node, and if something is happening to that node, suppose if that is a protein, and if that protein is not functioning well, so that may not be affecting, or that may not be this impacting the network. So only that would be at the corner of the network, that would not be impacting that much, impacting the network. But if the perturbation is causing or happening to the central node, that hub node, the entire network will get perturbed. So identifying the hub gene, that is an important aspect of this network pharmacology. And in Cytoscape itself, there are many algorithmic tools available, like this CytoHubba, CytoNCA, NCC. So those are the various apps or tools available in the Cytoscape software to identify the hub targets. So once we get the hub targets, that also we would be getting a list of hub targets. So then we need to do the functional enrichment. So functional enrichment means actually, so we need to know so what these targets are, these hub targets are doing in that disease case, or how important that hub nodes are in terms of the diseases we are dealing with. So whether that is acting on some key pathways or key biological process. To know that, we need to perform the functional enrichment analysis. So there are two types of this functional enrichment analysis. The first one is the Go enrichment analysis. So there are three key terms we analyze under Go, that is biological process, molecular function, and cellular component. Then the second one is the pathway analysis. So that means that what are the pathways in which these hub genes are important, or hub genes as well as the interacting partners of those hub genes are important. So that will give us that insight on so whether these hub genes are very much associated with the disease which is under study, and what are the pathways to be focused. So for getting that information, this enrichment analysis is essential. Then, so actually, as we are discussing this in a step-by-step manner. So, when we started from with the compounds, the targets, the all information we have gathered from the databases. So we know that this databases, that is also not a comprehensive this source, because the information present on the database, so that may not be complete. So based on the existing information, we are building this all these steps, and because of that, so when we get the output, so it is very much essential that we need to validate those outputs. So there are multiple ways of validation also. We can do in silico validation, in vitro validation, in vivo validation. So how we can do the in silico validation? So by doing the molecular docking, this assessment, we can do the in silico validation. For example, so after that enrichment analysis, what we would be getting is this list of compounds, list of targets, enriched pathways for that particular disease. So in order to see whether those compounds are actually modifying those targets, we can do this in silico molecular docking validation. So that gives us whether that compound is binding at the binding pockets of the targets, and whether that is that binding energy is this proper binding energy, kind of this good binding energy. So that is giving a kind of a good interaction between the target and the drug. So this is about the complete workflow of network pharmacology. Now let us have a recap of the workflow which we discussed right now. So we can start with any kind of this Ayurvedic formulation, or any kind of, even we can start with a botanical or combination of botanical. So what is your this study area? Based on that, you can decide. Even a few compounds also you can start with. Then the disease of your interest. So if this is your this approach, that is you want to see whether the formulation you have taken, that is good for this modulating this disease, and how. So that is your research question. So we can follow this kind of a research workflow, the network pharmacology workflow. So here you have to get the bioactive information, and this when we have to get the bioactive or compound information, and it is this good idea to go ahead with the metabolite profiling, that is LCMS or GCMS profiling, rather than going only with the databases or literature evidence, because the information present in the published literature, that would be very limited. So in order to have a very firm this information that okay, your the starting compound or formulation is having these metabolites or bioactives, you can go ahead with the this GCMS or LCMS analysis. So then you can find the targets for each of those bioactives. Parallelly, you can get the targets of the disease of your interest from various biological databases. Then you can find the common targets between the bioactive targets and the disease targets. Then you can do the prepare the bioactive target network. Also, you can construct the PPI network, that is the protein-protein interaction network. Then you can go ahead with the functional enrichment analysis, and pathway enrichment and Go enrichment can be performed. Then the validation, as we discussed, can be done through in silico molecular docking approach, or this in vitro cell culture approach, or in vivo experiments. So this is the complete workflow of the conventional. Right now we are calling it as conventional network pharmacology. So that is the workflow of this conventional network pharmacology. Hope this workflow is clear to everyone. Now, so let us look at the limitations of this conventional network pharmacology workflow. So, so far we discussed that kind of a systematic approach and this consolidated workflow and all, but that has some challenges or some limitations. So we are discussing four major limitations here. The first one is that is providing these networks which we are building through the conventional approach, that is giving us only a static knowledge. So what does that mean? So that is, we are building this network based on the existing information. So this is like, actually we are looking at the map also, that is kind of a this physical map. The earlier we used to this depend on such kind of physical map before the Google map, right? Google map era. So when we look at that one, actually we know that actually through this route we can reach to this place. So and we can follow only that route. So similarly, when we do construct the networks based on this, so that is giving based on the existing information only. So whether there is any changes happened after that network creation, that won't be reflecting in that network. And second one is this database dependency. So as we discussed, everything, so most of the information we are gathering from this databases or literature. So that means that if the information is correct, then okay, there is no problem. But if we are using some this irrelevant databases or irrelevant sources, the information we are gathering from that would be this biased or wrong information. So the network we are this constructing and the inference we are getting from that also would go wrong. So there is a lot of database dependency in the current conventional approach. Then so high false positives. So we are predicting the targets. So when we predict the targets, so that may not be, even though we have this many steps after that to validate those associations, but the predictions, so all the predictions, compound target predictions when we perform, so there is possibility of high false positives. And the limited learning capability. So as I told you, the static network, right? So if there is a new information that is not getting added, and every time, so for example, this plant A or formulation A, we are constructing a network. And if we have to go for the next formulation, we have to start from the beginning. So we cannot add to that network. So this network is not learning from that one or from the this additional information we are providing. So that kind of a limited learning capability is there for the conventional network pharmacology. Now, so let us see how this AI integration in AI integration can be done in this network pharmacology. So where does it fit? So where can we integrate this AI in network pharmacology? If we can integrate, where does this integration fit? So if we look at our network pharmacology process from starting from the data mining to this mechanism prediction, so in each of the stages we can integrate this AI, artificial intelligence. For example, in this starting from the data mining. So how did we do in the conventional approach? We dependent on the databases, but using AI, so we can source or mine data from this different sources, even structured as well as unstructured sources. We can this mine data from using this AI, artificial intelligence, and knowledge extraction. So from like this, there are a lot of hidden information, and this AI can read lot of this text, whatever text available. So AI can read information from that, and they can give us that as a knowledge or connection links. And for target prediction, so by this constructing this knowledge graphs, so they can give us this predictions, and they can also give us information regarding some hidden targets. And also this network learning. So if there is some new information or some additional information that is to be integrated, so we can integrate that to the existing one, and from that also this using AI tools, this they can learn information, and they can generate or synthesize new information. And they can also predict the mechanism. So they would be saying that, okay, this bioactive is this has an interaction or potential interaction with this target. So that AI, using this AI, that is not only predicting, that is having an interaction with that one, rather predicting, okay, so this is interacting with this target and this is through this pathway and this would be giving this kind of an effect. That's that kind of a this full connection the AI would be able to provide. And this beyond that, so we this AI also can help us or contribute in drug prioritization. So how do we do in conventional network pharmacology? So we identify the hub target, then we evaluate the hub target based on the scientific evidence. But here, using this AI, we can get the drug prioritization based on various parameters like drug efficacy score, then safety score, network, this topological parameters, ADMET score. This based on various scores, they can systematically give us a drug prioritization, and this prioritized list can be generated based on that. Then finally, the lead optimization. Sometimes what happens is that, so when we this find that, okay, this is a compound, a phytochemical that is suitable for targeting these multiple targets, and this is a proper lead. But when it comes to this biological this testing, so that may fail. So in that case, some fine tuning would be required on that lead. So that kind of a this information, that is this AI can also help us this provide analogs. So if that lead compound is not suitable, so which analog of that compound would be suitable? So that kind of an information also can be given by AI. So we this discussed a lot AI, and AI. So this AI means artificial intelligence. That is a very broad term. You know that, right? So let us see. So what are the this AI technologies? Those are actually helpful in this network pharmacology. So these are the this 8 such models I have listed over here. The first one is the machine learning. So you all would be knowing about the machine learning. So there, so they do the model, they do the prediction based on the models they build. That is based on the known data, they build the models, and when the unknown data point is given, so then the prediction would be happening. So that is how this machine learning models work. So this also helps in this our network pharmacology for this activity prediction, ADME prediction, QSR prediction, like that. And the second one is the deep learning. So you know that actually AlphaFold, that was a revolution in this biology. So this Google DeepMind's AlphaFold prediction, structure predictions. Now for each and every protein, so we can have the structure available. So that is mainly by this deep learning approaches. So this is by neural networks, and so they analyze that multiple layers of this network, and they just generate new information from that one. That is the deep learning approaches. So this helps us in this protein structure prediction, drug response prediction, and other analysis like time series analysis. And the third one is the graph neural networks, that is GNNs. These GNNs are very much important in terms of this network pharmacology. That is, when we construct a network, so in order to this identify the targets, so sometimes actually that target won't be a known target, but actually based on this network analysis, this using this GNN, that can identify this new connections. For example, so this is like our social, how our social media also works. That is actually, so in the Facebook, suppose, so one person would be having some this 50 mutual friends with the person, the next person. So the algorithm will give us a prediction. So you may know this person. So that is based on that this mutual connections. So likewise, in this network, our this biological network also, or this our drug target pathway network also, by analyzing the this neighboring targets, and they can identify using the graph neural networks models, they can identify the hidden targets information. So that is also a very good model that can be utilized in network pharmacology. Then the fourth one is the large language models. So here in network pharmacology, when we do that, actually, even though there are a lot of information available in the literature, so we may not be exploring those literature information because that is a time consuming process. But using this large language models, such information can be gathered, and so that is also in a faster this pace that can be gathered in a few minutes. We can gather. For example, if you want to know the reported targets for a this particular compound that is present in the literature, that may not be present in the databases, but that would be available in the literature. So we can gather that information through large language models, and that unstructured information they would be giving in a structured format. So, and then knowledge graphs. This knowledge graphs is a key component that we will discuss in the coming slide. Then generative AI. So once the pathway information that is also this developed, then if there are any connections we can make, for example, that compound is acting on a particular pathway and that impacting in a particular mode this disease. So if that kind of an information is there in any kind of this source or unstructured data, this generative AI is capable of getting that information. And then foundation models, those are pre-trained models that also can be useful in network pharmacology. And finally, the digital twins. So these are the recent advancements in this AI. So this digital twins means this virtual replicas of this biological systems. So that means that if we have to study this a patient, so we can make replicas of that one, and we can see which medicine would be more effective in that person. So mainly these digital twins are being utilized in this clinical trials and personalized medicine, and also for disease modeling. Okay, so we discussed about the this AI landscape, different AI models that can be utilized in network pharmacology. Now, let us look at the AI network pharmacology architecture. So with this, how this AI is helping in the network pharmacology and what kind of an information that can give. So that is from this knowledge and literature layer to this up to that clinical layer. This can give us the information. So this integrate all these layers like knowledge and literature layer, chemical and natural layer, biological network layer, omics layer, and clinical layer, and they can integrate all these together. And that is the this best part, or that is the very advantageous part this AI can do. Because look at these things, we know that there are a lot of heterogeneous data. So compound data that is different from the target data, that is different from the pathway data, likewise. So this AI can integrate all this heterogeneous data, and they can give us output like the target prediction, network inference, drug prioritization, and mechanism prediction. So this is kind of this integrated this architecture of this AI. Now let us look at the knowledge graph. Actually, this knowledge graph, so that is the core of this AI network pharmacology. That is for AI to predict something, or for AI to this understand or give us some information, we need to give AI that information in a particular way. So that is through this knowledge graph. That is various heterogeneous information we have to input in the AI models. So starting from plant, compound, likewise, then we need to have this integrated these many information. So this we are integrating in a particular this node and edge way. That is actually we are providing this information that this compound is interacting with this protein, likewise. And also actually there is no need for us to give this compound target interaction directly, so in a manual way. So we can say that you can explore these databases, that is whatever databases we want to integrate together, like phytochemical databases, gene databases, disease databases, all these databases together we can input in AI, and we can ask that to make a knowledge graph from this one. So they connect all this information together, and they make a network like this, and from this network they make the predictions. So that is the knowledge graph. So this knowledge graph, that is the key in AI in network pharmacology. And the next one is the graph neural network. As I told you, this GNN, that is also very important So that is through that one, this AI is learning the biological network. So here in traditional this network pharmacology, what we do is that we create network, we measure the this topological parameters, and we find the hub genes. But in AI network pharmacology, so based on this data input or structured and unstructured data input, this knowledge graphs have been created, and from that one, this learn the hidden topology. So in through GNN, they learn the topology, and they also they understand the all interactions, and they predict the hidden interaction, and they rank the such kind of this interaction according to their biological importance. So that is how GNN works in this networks knowledge graphs. So this is an example, that is the kind of a hypothetical example how this artificial intelligence network pharmacology work with this graph neural network. For example, so we initially we would be giving this linear this information, that is quercetin is acting with so and so targets. Then this AI builds that network. Then they analyze, actually then they learn that hidden topology, and they predict the unknown interaction. So in the they can visualize in that Google Map way also. So you would be this going to this one destination for this past many years. But right now, if you are using the Google Map, you know that that would be showing you a different route if the traffic congestion is high in the area which you are going. So so that would be kind of a very new route for you. So likewise here also, based on the lot of information in the network, and based on the hidden topology, they predict some unknown targets for the this compounds which we have provided. So that would be kind of a new information. So such kind of an exploration won't be possible in the conventional network pharmacology. And they do not predict only, but they link that prediction to this various ways, so that there would be kind of a this rational for us to believe that kind of a connection. So now actually I think you have got an idea about all these things, and we have only 3 minutes left also, so we can quickly discuss the applications of this AI. So this AI in network pharmacology can be used, as we discussed, for target prediction, AI-driven disease module identification, then for explainable AI for mechanism discovery, and for this can also be used for multi-state integration. Then this is a practical workflow for this phytochemical drug discovery. So like we discussed the workflow previously, here after this compound identification, we can build the knowledge graph. So from this knowledge graph, that is after that one, we can integrate all the databases together, and we can build the knowledge graph. And from the knowledge graph, this target prediction by AI can be possible. Then so we can analyze that network through this graph neural network, and disease module predictions are possible. That means that actually rather than identifying this those hub targets, so whether this AI analyzes whether there are some disease modules in that network. So then what is the importance of those disease modules? Then what is the mechanism through which that disease modules are modulated through this various compound and drugs? Then for this, then we can, after getting this kind of this information up to this mechanism prediction, we can go ahead with the molecular docking, molecular dynamics, and for ADME prediction also we can utilize the AI tools. Then for lead prioritization, and we can use the AI tool as we discussed previously. Then finally we can go ahead with the experimental validation. But we cannot eliminate experimental validation even though we are utilizing AI. So that is a compulsory part. So here this AI, with the AI also we are getting more information, more insights. So but finally we have to validate that through experimental ways. So various models that act at different stages of this workflow, that is for compound annotation, we can use LLM NLP models. For target identification, we can use GNN, this graph neural networks, and network analysis, we can use graph learning. For mechanism delivery, explainable AI. For ADMET prediction, deep learning, and for lead optimization, generative AI can be used. So even though we discussed a lot about this AI in network pharmacology, so there is a lot of limitations also. We know that that is a this is AI is an emerging area. So a lot of refinements has happened, and it is happening also. So after 10 years, so there would be kind of a more robust this information we can gather. Not there is, I think there is no need to wait up to 10 years. Every year by year, a lot of this new things or developments are happening. Okay, so but let us look at these key challenges of this AI in network pharmacology also. The main thing is the data dependency. So like we discussed about the conventional or classic network pharmacology, here also this data dependency is there. So what we are giving as the input, so here also we are giving the database information as the input. So this is getting information from that databases. If that is not correct, or that is not in this proper way, so AI cannot this make good predictions. So that data part, that input part, that is to be this proper. Then data quality and bias. That means that actually the bias means, so for example, there will be some this compounds like quercetin, this and curcumin and all. So those are highly explored compounds. So there will be a lot of target information for such compounds. But there are some other compounds so that are not being explored this that much. So the target information would be limited for that. So when we develop a network, we would be getting a very small network for such compounds. So that does not mean that for that compound only that is there, but the information that much is there. That is why that network is very small network. That kind of data bias is also there. That is one of the challenges in AI. Then lack of interpretability. In some cases, this AI predict that okay, this is a good target, this is an unknown target, but this is a good target. But sometimes they would not be able to provide a scientific justification for that. So that time we call that as a black box model. So because that is not clearly explained. So that kind of an issue is there in AI. And as we discussed, the experimental validation that is required. All this would be coming as a prediction or this in silico information. We need to experimentally validate all those things. So AI accelerates discovery, but it does not replace biological validation. But on this moving on, so actually there will be a lot of this changes coming, lot of developments coming in terms of this AI and network pharmacology. So this AI would be this transforming network pharmacology from this descriptive analysis to the predictive intelligence. And another thing is that, so we know that there are a lot of this omics information like transcriptomics, genomics, proteomics, metabolomics, those information that is available. So in this network, what we are preparing, so we can integrate such omics information and we can utilize AI to get inference from that kind of a network. So then continuous learning. So when the information we are getting, like in Wikipedia, so we can get this recent updates also, right? So likewise, so in this one also we can do the continuous learning, so that enables smarter target and drug prioritization. Then this AI-driven network pharmacology, this can also accelerate the precision phytochemical drug discovery. So that means that, so one formulation that won't be fitting for all. So which formulation or which this phytochemical drug that is good for a person compared to another person. So that kind of an intelligence or this or precision medicine approach can be attained using this AI, artificial intelligence in network pharmacology. So we can hope that actually a lot of developments also coming in the future. But so we actually it is sure that so there are a lot of this progressive developments happening, and the future of this AI in network pharmacology, that is definitely a bright one. So thank you for your this patient listening. So, and if you wish to join our this WhatsApp community, that is the Scinome's WhatsApp community, you can scan this QR code, and so we update our this trainings and our this news information, everything in the WhatsApp group. So if you wish to join, please join. And this is our website information, www.scinome.com. And if you have any this question, or if you want to know anything about us, you can WhatsApp us, or you can follow our LinkedIn page. Thank you so much for your patient listening.
Thank you so much, ma'am. Okay, there are a few questions in the chat box. Can you see them?
Let me check, sir.
Not, I will read up. One, we'll start from the top. Few will take.
Yeah, sure.
What are the limitation of network pharmacology?
That we have already discussed, sir.
Correct. What is evolution optimized molecules?
Yeah, so that means that this in plants actually based on so for example, as I told you, the plants cannot move from one position to another. So for them to survive, so they have to this withstand all the this surroundings, and whatever attacks they are getting from the surroundings. For that, they are developing secondary metabolites in them. So that is evolution optimized molecules. So that is what those are known as the secondary metabolites in plants.
Okay, correct. Can't I use network pharmacology for approved compounds or new chemical entities? I think you have already covered this also.
Yeah, yes, we can use network pharmacology for approved compounds or new chemical entities. If that is a compound, that is, we are building from this chemistry to this biology. So if that is a chemical compound, yes, definitely you can go ahead with plan B.
Okay. Next question I didn't get actually. Real biological system interactions like a temporal changes, feedback loops, and tissue-specific variations that are captured in network pharmacology.
Yeah, that also we discussed. So we're
Okay,
utilizing AI, we can this address that kind of a limitation.
correct. Okay. Can you give a list of tools that can be used for target prediction?
I discussed that too.
Okay, correct. Okay. Okay, yes. One thing, how are nanotechnology and phytochemicals being combined for drug delivery? I guess this.
That is out of the scope of this one, but I think you can explore that.
Okay.
That is for drug delivery.
Okay. AI tools you have already mentioned, right? Which
Yes,
AI tools can be used for integration?
yes.
How does the lack of standardized workflows affect reproducibility in network pharmacology?
Yeah, so that affects a lot in network pharmacology. Many people they just do not follow the standardized workflow, and so that results in this less quality publications. But whenever you do network pharmacology, follow a proper this workflow.
Okay. One more question from academic background is that what foundational software tools, open source programming package or core competency do you recommend? We introduce to our students so that
Yeah.
they are equipped with AI-driven R&D workforce.
For AI driven or network pharmacology, I have already discussed the tools and everything. For AI also, I have discussed the models which can be used.
Can be given to the students also.
Yeah, yes, they can explore the knowledge, a generative AI, knowledge graphs, GNNs. If they can explore, yes, definitely they can explore them.
Correct. Some compounds are not listed in databases, which was isolated by our own friend. How can this limitation be overcome?
Yeah, so that is what I explained. So there is whenever we use this database, whatever we are doing using network pharmacology, that is based on the existing information. So if you are identifying a new compound, you can this publish a paper using that one, this using proper evidence and all. So then another person, the next person can utilize this compound. So that is how this is growing.
Okay. One more question is there. Is it possible to conduct a network pharmacology study on a synthetic drug?
Yeah, that is possible.
Okay. Can you share that WhatsApp link that you shared? Someone they are asking
Yeah, sure.
screen.
Yes, I'll share. Can you see?
Yes. One more question. Can we take?
Yeah, sure.
Can we perform network pharmacology specific metabolic pathways from the disease rather than selecting every target?
Yeah, yes, we can do that. Whatever is your research question, based on that, you can develop a network pharmacology workflow. or you can just fine tune, refine the network pharmacology workflow in that way.
What is the scientific rationale for using it to study a synthetic drug, network pharmacology?
So that is to see whether there are this your synthetic compound is acting on multiple targets and how those are modulating different pathways.
Okay. With respect to the AI tools, the same question is coming up. Madam had already discussed that. One more question was Swiss target prediction. Should I
Mhm.
take targets for non-zero probability score?
No, that is not a good idea. So the when you take the use the Swiss target prediction, use a proper cutoff. Otherwise, if you are taking that one, then you need to validate that properly. So that is again a this tedious task. But ensure that if you suppose if you are planning to do some wet lab experiments, then in that case you can take. Only thing is that you have to confirm by doing the experiments.
Okay, with respect to the Zoho IDs, the IDs that are displayed on your screen are your Zoho IDs. So that's not an issue. Any ID that will help us in identifying you. That's the only thing. Okay, so we'll stop here, ma'am.
Yeah, sure.
Okay. Thank you very much, ma'am, for such a wonderful, insightful, and informative session. We really appreciate the time that you have taken to share your knowledge and also the valuable experience with all of us. I'm very sure that today's session or the discussion have provided our participants with good perspectives and also practical insights in this very interesting field, which is also very upcoming with respect to the AI tools and all. I would like to extend sincere thanks to the participants also. They have most of them have stayed back. We have almost crossed that 100 mark. We had set that mark because of the limitation of the app, and they, of course, the questions themselves show that they are very active participants. It is because of this enthusiasm and the engagement that we have that makes this knowledge series a very meaningful platform for learning and also with respect to professional growth. Before we conclude, I would just want to inform that the Google form link has already been shared in the chat box. Kindly fill it so that we can issue the participant certificates. Once again, I'm really thankful to each one of you for this 20-second session of this Spectratix Knowledge Series. Special thanks to ma'am. Anything from your side, ma'am, before we conclude?
Yeah, actually, thank you so much. Actually, thanks to the Spectratix for also this conducting such webinar sessions. So I think that is helpful for many. So by seeing the participant number, also I can make out that actually. So that is helping a lot of people. So thank you for this opportunity. And if you have any of the participants, if you wish to explore this network pharmacology further, please reach out to us. Thank you.
Okay. Thank you so much to each one of you. Thank you. We'll see you in the next session. Thank you. Bye.
Thank you. Bye.